Hi, thank you for that wonderful introduction, and I'm really pleased to be here. I first want to start off by thanking Rob Jackson and Michael Brockhurst for the nomination for this prize and to the committee for the award. I feel very honored and excited to be here today to talk to you about the challenges and opportunities in understanding the factors shaping the plant microbiome. So since I've been a scientist, I've been trying to explain diversity. How is it generated? How is it maintained over time? Because you need only look out your window to realize how incredible it is in terms of how many species you can see, let alone all of those taxa that you can't see. And when I, for my postdoctoral work, moved into microbiology and the worlds of microbiomes, I took all of the same principles that I had been thinking about in terms of plant and animal communities to explain this incredible diversity. And so the existing fields of ecology and evolution are ripe with amazing theories and empirical tests of mechanisms by which diversity is maintained. And much of what I'm interested in the moment is how many of these ideas translate over to the microbiome, and in which ways is the microbiome truly unique. And so I think about the many factors shaping the microbiome, and I'm going to talk in today's talk about 4 of them. I'm going to sort of do a ride around the carousel and give you little vignettes of each one. And the first factor I think about is the environment. So for example, how does the local pH and temperature or shade affect the microbes that are found there? And the other part of the environment, of course, that's important is the amount of resources that are there, the types of resources as well. And on that point, the second factor that we need to take into consideration is competition and cooperation, so within microbiome interactions. And that can include direct competition for resources or indirect competition through toxin and antibiotic production. The third one is, just like plant and animal communities, the role that parasites and predators play in shaping these communities. And for today's talk, the parasite or predator that I'll be talking about are bacteriophage viruses that infect and kill bacterial cells. And then the one point where we start to really diverge from plant and animal communities is the role that the host plays in shaping host-associated microbiomes. And so this could be, for example, a result of long history of coevolution between a given host population and its microbiome. It could simply be a host filtering effect by which the different host genotypes and the exudate production, for example, of one host changes which microbes can colonize that host relative to a different host. Or it could also be the result of bacterial adaptation to particular host traits. And so, as I said, we'll do a ride around this carousel today with short vignettes from each one, just to give you an idea of how we test these ideas. And I'm going to be doing this focused on the Phyllosphere. So when you think about the plant microbiome, you might often think about below ground, the rhizosphere microbiome, all of those bacteria, fungi, and viruses that are associated with roots. And this is obviously very important for plant health. Indeed, if you do a Google Scholar search for rhizosphere and plant health, you get over 100,000 hits. But I actually am going to be focusing above ground on the Phyllosphere. And here, there are increasingly recognized benefits of the Phyllosphere to plant health, but much less well studied. So a little over 15,000 publications currently in terms of Phyllosphere and plant health. But for me, the reason I think the Phyllosphere is a fantastic model system is because it's spatially explicit. So as an ecologist, I really understand where populations and communities start and end. It's also, as I'll show you through today's talk, it's easy to measure and easy to manipulate. And as I said, we're increasingly recognizing the fact that it's important in terms of plant health. Then the final thing is in terms of just a habitat for bacteria. The plant Phyllosphere makes up about 60% of the biomass on Earth. And so this is an incredibly rich habitat in which to be adapted to be able to live and thrive. Okay, so let's start our tour here. We're going to start, as I said, with the environment. And this is some of the newer work out of our lab, which is in prep for publication right now. And the idea here was to ask how below ground environment shapes this above ground Phyllosphere microbiome. And the work was led by graduate student Rena DeBray, and it was in collaboration with Tim Bowles here at UC Berkeley and his graduate student Yvonne Sokolar. And Yvonne and Tim set up a fantastic field trial at UC Davis where they asked the question of how plant growth and fitness is affected by below ground factors. And the factors they worked on are water availability, so drought, as well as phosphorus in the soil. And then the third one was below ground associations with mycorrhizae. And in this case, they used a wild type tomato plant and a tomato plant that is deficient in its ability to form associations with AMF. And this, for us, led to an exciting opportunity to ask how these below ground factors change the above ground Phyllosphere microbiome. And the first thing I'm going to show you is just that it does. And so this is a classic sort of a principal components test where you can take a very complicated data set like a microbiome with that rich taxonomic diversity and fit it onto two axes. And this is simply a way of visualizing how different two communities are from one another. So if the points are close together, those are very similar communities, and if they're far apart, those are very different communities. And all I want you to really take away from this is relative to wild type tomato that are grown in full water, which is this yellow circle, all of the other perturbations, all the other stress we put them under led to a shift away from that sort of wild type, if you will, microbiome. But the second thing I want you to take away is what impact this had on diversity. And in microbiomes in particular, a general rule of thumb, although there are many exceptions, is that higher diversity of the microbiome is associated with better health, with better function. And in this case, what we find is the highest alpha diversity, so species richness, over on the left here with the wild type tomato plants that were grown in full water. And if you start stressing those plants out, putting them into drought or reducing their below ground associations with mycorrhizae, then you lose diversity. The other nice thing about this is you can look at specifically who is lost and who is gained. And so in the purple here, under the water regime, so with high water on the right and low water on the left, or in terms of mycorrhizal association with the wild type on the right and the mutant on the left, we see amazingly parallel shifts in who's enriched in wild type and water in the purple and which taxa are depleted. So in other words, they're found more under drought or in the mutant type. And so I don't want to spend much time here in terms of why this is the case, but I do want to emphasize here that this is very clear evidence that certain taxa are shaped in their presence and absence above ground, as well as community diversity overall, by what's happening below ground. And we think of that below ground as the environment that's shaping the plant growth and fitness. Okay, so continuing our ride around the carousel, the next thing I want to talk about is competition. And I will acknowledge that cooperation and cross-feeding is critically important in the microbiome, but for today's talk, I'm going to specifically think about competition. And the type of competition that we're most excited about at the moment is competition between an arriving pathogen and the resident microbiome. And this is the work I'm presenting here is led by Shirley Zhang and Norma Morella. So Shirley was a fantastic undergraduate researcher in the lab who is now doing her PhD at Cornell on the East Coast, and Norma Morella was a graduate student at the time who's now a postdoctoral fellow at the Fred Hutch Institute. And they were interested in the role of the seed-associated microbiome in protecting against pathogens. Okay, and so what do I mean by that? I basically mean that we've known for a long time that seeds have epiphytes. They have a microbial community associated with them. And so Shirley and Norma took advantage of this to do the sort of plant equivalent of a fecal microbiome transplant. All right, so they collected tomato from 4 different types of tomato in the field, growing in sort of natural agricultural settings. They brought them into the lab, surface sterilized the tomatoes, and then fermented the fruit just like you would normally to collect seeds, collected the seeds from those 4 different types of tomato, and then used a jewelry sonicator to gently shake off and disassociate the epiphytic microbiome of the seeds from the seeds themselves. They collected the epiphytic microbiome and then they surface sterilized the seeds, so to rid them of any remaining bacteria, and then germinated those seeds in water agar under sterile conditions, either with a re-inoculation of the resident microbiome or without the resident microbiome. And then once the seedlings had germinated, they challenged those seedlings with a plant pathogen, Pseudomonas syringae, which causes bacterial speck. and then followed disease progression over time. And this is what that looks like. So on the y-axis, we have time as the seedlings are growing and maturing and becoming diseased. And on the y-axis, we have disease severity. And there's a picture of what bacterial speck looks like on these seedlings on the right. And what we found is that on average, and certainly in tomato types 1, 3, and 4, there was a very protective effect of the resident microbiome. In other words, you saw more disease, so higher points, when these seeds received only buffer than when they were re-inoculated with their resident microbiome. And this is exciting, and it matched some of the work we had done previously in adult plants, so the above ground leaves of adult plants. But Shirley didn't stop there. She sequenced all of these microbiomes to look at their diversity and discovered that they're not very diverse, these seeds, but that they were mainly Pantoia. And she then went in and isolated individual Pantoia species from these microbiomes, from these seeds, and asked a simple question, which is, is one of these taxa enough to confer this protection? Or do you need a consortia, for example? And in this particular case, these ZM1, ZM2, and ZM3 are the Pantoia isolates, the novel species that she isolated from the seeds. And you can see, first we have them alone inoculated onto seeds, and then in consortia in the middle here, and then C91 and these other two are biocontrol strains that are on the market. And importantly, on the far right is no treatment. So these were seeds that were germinated without a resident microbiome. Now on the y-axis, this is slightly different. We're looking at area under the disease progression curve. So if you imagine the curves I just showed you, this is now calculating the area under there. So higher area means more disease throughout the disease progression curve, and lower area is less disease. And what you can see is just single isolates are enough to confer this incredible protection. And actually the consortia, in some cases, even performed worse than single strains. That's on terms of disease progression, and then on the right we use droplet digital PCR to measure absolute abundance of the pathogen. And again, these single strains, as well as the consortia, were enough to really stop the pathogen from growing. And this told us that there was a direct competitive effect, but we also, I won't show the data here, but Shirley also used heat-killed inocula, heat-killed strains, and inoculated them onto seeds, and could show that in that case there was also protection, although not as much as we observed with the live bacteria. And that tells us two things. It tells us that live bacteria are better at preventing the pathogen from growing, so this is probably through direct competition, but also that the plant is somehow mediating this. So something about the defensive symbiont mediating defense in the plant. So you still see a bit of this defense even if the bacteria is not alive. Okay, so most of what we do in my lab is actually focused on these two top ones, so I'm going to spend a little bit longer here. And I'm going to start with phages and tell you two short vignettes about phage. And the first one is a simple story about how phages might shape disease progression. So leading off of what we just saw in terms of defensive symbionts, now moving from competitors to parasites. And this is work that was led by Kathy Hernandez, a graduate student in my lab, and a summer student, fellowship student, Andrea Salazar. And what they did is they took a phage that we use in the lab quite often, and it's a lytic bacteriophage, so it binds to bacterial cells, injects its own DNA into the cell, hijacks the cell replication machinery, and the cell now reproduces phage particles, at which point it lyses the cells open, and those phages are released into the environment to infect new bacteria. And so the idea, which has been around a very long time, since the 20s, is that these phages as bacterial killers could be used to reduce disease. And we tested that in this seedling system that I just introduced. And so I'm showing you just one result, one set of results, where there's a lot more in the paper. But this is our straightforward result here, which is that if you're now, again, this is a disease progression curve, just like you've seen from 7 days post-inoculation with Pseudomonas syringae. And this is just Pseudomonas syringae on the top, and you see that it indeed causes disease. But if you combine it with, this is phage 1 here in the blue, sorry. If you combine the bacteria with phage, that phage reduces the bacterial density and also disease. And of course, this is two controls here. This is the phage alone. We do not see any disease. And this is a magnesium chloride control here in gray. And on the right, we use droplet digital PCR to measure absolute abundance of the pathogen, but we can also measure absolute abundance of the phage. And so when you look at bacteria only, what this is showing is there's a lot of bacteria and no phage. When you combine bacteria and phage, there's less bacteria, that's that blue open bar here, and a lot of phage, blue shaded gray bar. And if you have only phage, there's no reproductive capability, so the phage does not grow. Okay, so this is just one example. There's many pieces of evidence like they're out there. This is one example of how phages can target a specific bacterial population, in this case a pathogen. But what role do phages play in the microbiome more broadly? And this is work that was led again by Norma Morella, who I've already introduced, and a wonderful Amgen scholar at the time, Annika Gomez. And so what they did was ask this question of the role that phages play in maintaining diversity in the microbiome as a whole. And they took advantage of a method that's been well developed in marine systems for marine phage, where you take a cup of seawater, or in our case, a cup of leaf wash, where we went to agricultural settings, collected the Phyllosphere of field-grown tomato plants, sonicated off all of the microbes above ground, much like we did with the seeds, but at the whole plant level, and then filtered that to collect the bacterial communities on top of the filter. So this is what gets caught are the bacterial and fungal communities. And then what goes through the filtrate, we used to concentrate viral-like particles using this 100 kilodalton filter. What this then meant is we were able to separate the community into bacteria that was depleted of viruses, not necessarily entirely absent, but mostly absent, and the phage. And then we could spray juvenile, mostly sterile tomato plants in the growth chamber with either the bacteria alone or the bacteria recombined with its phage community, and use droplet digital PCR as well as 16S amplicon sequencing to measure both the abundance of bacteria, what these phages did to overall numbers, and what the phages did to diversity. So I'm showing all the data at once, but I'll walk you through it slowly. So if we start on the left here, this is our result of droplet digital PCR to measure overall abundance of the whole community. So we used a 16S probe that will tell us how many bacterial cells, independent of taxa, are on the leaf. And what we found is that if you spray the same inoculum, but just the bacterial component of it, you get a lot of growth during that first day of colonization. But if you recombine that bacteria, that same inoculum, but with its phage, then you see much reduced abundance. And this is telling us two things. It's telling us that these natural field-grown tomatoes had lytic bacteriophages, and those phages are capable of killing the bacterial community of individual members in there, most likely. Okay, so during that first day, we see a reduction in density. What does that do to diversity? And on the right here, the figure on the left, the alpha diversity measure, was using 16S amplicon profiling of the community. And what we did is a week in, so at day 7, we profiled the communities, and we could show that phages increased diversity overall. So there were more taxa found. There was higher richness found when the phages were recombined with the bacteria than when they weren't. And at first this might sound counterintuitive because I just told you abundance is lower. But actually, from a theory perspective, it makes perfect sense. And the idea dates back. It's the idea of parasite-mediated negative frequency-dependent selection, or as we call it in microbial ecology with a bit of nuance, is kill the winner hypothesis. And the idea is somewhat simple. It's that phages should be under selection pressure to target and infect the most common or the most competitive bacteria. Okay? And that means that the most otherwise dominant bacteria will be reduced in density in the presence of phage, and therefore that opens up niche space and resources for more rare taxa. And that's exactly what we found at this whole microbiome level. What we were maybe a little more surprised about is what it did to differences among plants. So what you're looking at on the right is the average Bray-Curtis distance between plants. What do I mean by that? I mean, 2 plants that were sprayed with the same inoculum, but just bacteria, how different are those microbiomes from one another after a week? And the answer is quite different, and much more different than plants that were sprayed with the same inoculum, but that was bacteria and phage. All right? And so this is sort of saying that phages have this ability to steer the microbiome in a particular direction and make hosts more similar to one another. And I think this is really exciting for the sort of future of phage therapy to think about it as a way to manipulate the microbiome. So we're on our final stop of the carousel, our tour around the factors shaping the microbiome. And here we're going to think about the role that the host plays. And really, for this experiment, we were interested in the role of the host as an environment to which the microbiome can adapt. And this is a huge tour de force effort led by Norma Morella in collaboration with Steve Lindau here at UC Berkeley and Jess Metcalf, who's at Princeton. And what we did was took a initially very diverse inoculum from the field, from tomato plants, and sprayed them onto replicate plants in the greenhouse over many plant generations. So let me show you what that looks like. So we started with a very diverse inoculum because we wanted a lot of standing variation upon which to select. So this is from different fields and different cultivars of tomato. We combined it into a single inoculum, though, and sprayed all replicate plants of 5 different tomato genotypes with this inoculum. We sprayed first starting at 2 weeks old, and then we sprayed weekly until week 7. And then at week 8, we harvested the plants, and we sonicated off all of the microbes from that above ground component of each line. And then that microbiome became the inoculum for the next plant in the next generation. Okay, so all of these lines were independent of one another. There was no mixing of microbes. We did that for 4 passages. And then at the end, we sequenced all passages. So we sequenced what the communities looked like at each point in time, as well as used droplet digital PCR to measure overall abundance of the communities. So the first question we had is, did we succeed in selecting on the microbiome? And how do we even know if we selected? And so I'm going to show you 2 pieces of evidence to how we convinced ourselves that we had indeed selected on this microbiome. If you focus first on the green dashed line here, what I'm showing you is the percent of the original inoculum taxa that was remaining on the plants at each passage. So we sequenced the original inoculum that we sprayed on the plants, so we knew what taxa we began selection with. And then we asked how many of them remained through time. And you can see that that drastically reduces through time. So by the end of the experiment, only 25% or so, 30%, of the original taxa that were there at the start of the experiment remained. So we clearly selected out about 75% of taxa. But then the question is, what are they replaced with? Things that are flying around the greenhouse, in which case that would be a little worrying. Or were those 25 really better? 25%. And so the bars here are showing you the percent of the bacteria community that we found on the plants at each generation that were from the original field inoculum. And so if you just focus on this final time point passage 4, I told you that only about 30% of taxa were remaining, but those 30% of taxa made up over 90% of the bacteria we found on the leaves. And so this really convinced us that we had successfully selected. But what did we do? And so this is another one of those principal component plots where you can see change through time. And far left here in the red is that first passage. And what we find in the first passage is that genotype, there were 5 different plant genotypes, explained about 30% of the variation in who colonized. So in other words, it mattered what the plant genotype was for which taxa settled there and thrived. The other thing to notice is that, of course, time matters. So as we move through to passage 2 in green, 3 in blue, and 4 in purple, the communities are shifting, and in a very concerted way. They're moving together away from that starting point. And so that tells us that even though these were independent selection lines, they're being selected in a similar direction. And also, interestingly, by the end of the experiment, the genotype of the plant no longer mattered. Okay. So that was great and really convinced us that we're moving in the right direction. We are moving these microbiomes in a way, but again, it doesn't tell us whether they are adapted. And so to test that, Norma ran what we call our community crashing experiment or community coalescence experiment. And what she did is she sprayed plants in the greenhouse with either the inoculum that came off of the plants from passage 1, so at the start of the experiment, or the inoculum that came off of the plants at passage 4, so our selected microbiome, or a 1-to-1 mix of live cells. So she used live dead droplet digital PCR to measure the actual number of live cells in each of our inocula and made a 1-to-1 mix. So what I'm showing you here is just the plants that received the sort of 2 parental inocula. And what you're seeing, this is the first time they were sprayed at the same time in the same greenhouse. And indeed, the plants separate out. So the P1 inocula plants have a very different community structure than P4. But what happened to the mix? That is the next question. Sorry, child interruption there. So I showed you the two sets of plants that received the two inocula. What happened to the one that received the mix? Well, the neutral assumption, the neutral model, would be that it was in the middle, right? Because it received a mix of both. But what we actually found is that it was statistically indistinguishable from the selected microbiome. And this is really exciting because it tells us that that selected microbiome is now uninvadable by those original taxa that we selected out. And so I think there's a huge amount of promise in this for many reasons, but in one way that it's telling us that we can select for microbiomes that are really well adapted to a particular environment. And we've yet to test what effects this has on plant fitness, but you could imagine that that's the next step. And then, of course, also trying to associate these microbiomes with functions of interest is a really exciting opportunity as well. So just to wrap up here, I've finished the ride around the carousel. I've shown you 4 examples of how these different factors can shape microbiome diversity. In terms of where we're going next, we're very interested in how these interactions have already been disrupted or might continue to be disrupted based on current agricultural practices. And again, once we have that understanding of how agricultural change has altered the interactions between the plant and the microbiome, we can then leverage this knowledge to create the next generation of agricultural sciences, where we have reduced reliance on chemical controls for disease and fertilization. And I think this is going to be a huge factor in increasing yield with less environmental impact, so moving towards more sustainable agriculture. But really what I want to end on is this. We spend a lot of time as microbial ecologists thinking about diversity in our microbial world, but of course this is just as important in our macrobial world, if you will. And so with microbiomes, diversity is power. When you have a diverse microbiome, that leads to incredible functionality and adaptability and just incredible benefits to the host in this case. And so much is the same with scientific teams and scientific communities. And so I'm not going to paraphrase this because I think Miranda Montgomery got this so right. I love this quote so much, so I'll just read it out loud. She was arguing in a recent publication that we can learn many lessons about equity and stewardship-based engagement from the ways that microbiologists seek to understand how to cultivate unculturable bacteria, including the importance of understanding an organism's language and community, replicating aspects of the environment of origin, an organism's occasional need to transform aspects of its environment to persist, and the critical needs to provide a range of culture conditions to support diverse organisms. And so moving forward as a society and as a microbiological community, I think it's absolutely critical to create a truly equitable, diverse, and inclusive community as a way to succeed in science. Thank you very much, and I look forward to the question and answer and any further questions you might have.